Hi, good morning. First of all, I would like to thank the organizing committee for the nice invitation. So I'm sharing my screen. So today I'm going to speak a little about biomarkers discovery. So I'm from Lisbon. You are all welcome. So what is the relevance of the precision medicine? So we want to discover biomarkers that can discriminate different types of diseases. And in this case, we could increase the efficiency of the therapies. For instance, breast cancer, we have different subtypes of breast cancer. Some of them will overexpress hormonal receptors, and the therapy must be to blockage these receptors. There are other types of breast cancer that overexpress other kind of receptors. So, and medical therapy must be different. And if you discover these biomarkers, you will be able to do this more efficient, more precise medicine. So, how to discover these biomarkers? We could conduct a bottom-up approach. So if you understand the mechanism of disease and if you discover something about the mechanism of disease on a characteristic of that disease, you will tell that you are targeting a biomarker. But if you don't understand how this is working, we could conduct a completely different strategy. We call it top-down strategy. And in this case, actually, we could compare different kinds of population, for instance, a health and a disease population, try to find the biomarker that discriminates these two populations. Actually, we call it is an on-target because we don't know what is our target, and it's an holistic strategy. So most of my I'm focused on this one. And based on both strategies, actually in the last 20 years, medicine has evolved a lot. So today, instead of we all reaching an amazing step in humanity, so because we are reaching a much bigger understanding of mechanisms of disease, and we are able to discover these biomarkers and more efficient therapies. So for this bottom-up strategy, we are going to compare a set of population that has this disease, a set of the population don't have this disease, and try to define with bioinformatical tools this subcharacterize. So, and how to analyze? Actually, on the last 20 years or so, technology evolved a lot. And we'll have technologies today that we are able to characterize the whole system considering the subtypes of molecules, genes, RNA, proteins, metabolites. So we are on the omics science era. So we call it genomics when we are looking for the whole genome of the system, of proteome or transcriptome or the metabolome. And for instance, at the beginning of the millennium, we achieved the first sequences of the human genome. And for that, the humanity took more or less 10 years, more than $1 billion. And we achieved this at the beginning of the millennium. Today, the cost to sequence a whole genome is much cheaper, and instead of the 10 years, we manage that with a high resolution on 2 days or less, depending on the resolution you want. So technology evolved a lot. And so we could conduct this omics, so try to characterize the whole systems, but we could check on genome or on the metabolites. Actually, I'm focused at the end of this scenario. Why? Because here is exactly what the cell is conducting on this precise moment. So actually, I'm focused on metabolomics. Focus on metabolomics. So we need to extract these small metabolites from the systems and then analyze it. And then with machine learning algorithms, try to find these different patterns between the disease and the health population. But what kind of the technique? Actually, we have available lots of different techniques to conduct this. Unfortunately, all of them are very complex, and unfortunately for us, each technique are looking for a different set of metabolites. Actually, none of these techniques are enable us to look to the whole system. So none of them are complete. Are all of them very time consuming, high labor. So for all of that reason, I'm also focused on a much simpler technique, that is Fourier transform infrared spectroscopy. On that technique, we are looking for vibrations between molecules. So we are also looking for the whole composition of the molecule of the system, but on a much cheaper, economic, and high throughput mode. So on the following slides, I'm going to show you some outputs that we achieve based on this technique, but also on a more classic metabolomic technique that using a liquid chromatography associated to high resolution mass spectrometry that is highly complex. So on this scenario, we are going to compare both platforms. So using the high performance liquid chromatography associated to high resolution mass spectrometry, highly complex, and we'll compare the data with the Fourier transform infrared spectroscopy. So we are comparing two groups of patients from the intensive care unit. So they are critical ill patients, and we'll try to compare both populations. So it's a small dimension population, and we'll try to predict the death of one of the groups. So both of the groups were under invasive mechanical ventilation, so they are very critical, but one of the groups actually die, unfortunately, and the other survive. So we want to understand, using these two platforms, if you are able to predict what kind of this group has a higher risk of death, so of dying. This is a very complex process because the metabolome is a very complex, huge quantity of molecules, and we are also comparing populations with a high variability. So it's very difficult to find a pattern and associate this to a metabolic state. For that, we use machine learning algorithms to find this a needle in the haystack, how you call it. So this is results. This is some of the metabolites that were statistically different between these two populations, the one that died and the other one that survived, and applying a linear discriminant analysis, that was one of the algorithms that we evaluated, we are able to predict with a high sensitivity around 80% and almost 90% specificity which patients were going to die. So it was a very nice outputs, and these outputs were based on the conventional metabolomic platform. So comparing this with the Fourier transform spectroscopy, we were able to manage also very high sensitivity specificities on obtaining on predicting which patients were dying. And this was very similar performance metrics. So this is a wonderful output. We conduct many of these kind of analysis using Fourier transform spectroscopy, for instance, to predict if a person presents genotoxic outputs on leukocytes on the blood. So this was based on a population that was unfortunately exposed, were hospital professionals exposed to chemotherapy reagents that induce genotoxic effects. So we have some population with the genotoxic already biomarkers, but these outputs took more or less 2 weeks to obtain. So we want to know if from a single drop of blood, we're able to predict this based on infrared spectroscopy. So this is the infrared spectroscopy of the human serum, obtained from a very small volume. And already, if you look from some of these bands, already managed some of these bands were very statistically different. A simple principal component analysis of the spectra of the serum already shows us that we're able to see a pattern different from the persons with these genotoxic biomarkers from the persons that didn't have it. And the actual support vector machines algorithms present very good performance to predict which person presents this genotoxic biomarker. So this was a wonderful output. We're able also to conduct the same kind of experiment, predict, to discriminate what kind of immune cells were activated. This is a very relevant information to many diseases. We are also able to predict this. And we also did some experimentation to predict the rejection of renal allograft in transplanted patients. And we conduct ISEL, a kind of experience in other system, always with a very nice output. Before presenting or finalizing my presentation, I'm going to show you data slightly different. This is from an image analysis using also Fourier transform infrared spectroscopy, but that we are able to acquire a pattern. So it's an image from a tissue biopsy from prostate. So we are using an array of detectors, actually 64 for 64 detectors. And this is a presentation from the average spectrum from this is prostate, so adenocarcinoma for hyperplasia, so it's not cancer still. And we managed to reconstruct this area under some of this band, and we have here the infrared images reconstructing almost the visual images that the clinicians were observing at the optical microscope. But we wanted to know if we're able to develop these predicting models. So we evaluated different kind of algorithms, all of these, and we managed to have a high accuracy comparing to the conventional outputs. We were able to predict which of these biopsy is associated to a cancer. So this is a very nice output also. So just to finish, the techniques, both platforms, the more conventional based on liquid chromatography associated to high resolution mass spectrometry, are very wonderful techniques. However, is very complex and labor, while the Fourier transform infrared spectroscopy is more simple, economic, and high throughput, so easily to translate to large scale studies, very relevant to achieve this. So you have this my contact if you have some questions, because today I'm not able to be here presential. Please contact me, would be a pleasure. Thank you very much.